I'm going to dark mode instead of bright light because bioinformatics, we like dark. So to do our analyses, we're really utilizing this Lima package that was developed for microarray experiments way back in the day when you'd have your gene expression and you'd go across different batches of different array experiments. And so this is a linear regression type model package that will perform all the statistics. But what it also does is it's running a moderated t-test that's going to calculate your variability within your samples. In the past, before the mass spectrometers got to where we could sequence 5,000, 10,000 proteins, when we were only doing a few hundred or maybe 1,000, Lima would not work because you need to have enough proteins within your data to actually estimate variability. So the faster and the better the machines have gotten, the more I'm able to actually pull from some of the resources on the genomic side for analysis, which has really kind of helped a lot. So Lima's is, I'm going to try to kind of go through high-level experimental design of your projects. And so there's no real good, bad. The bad are just going to be really complex and some things that we want to avoid. So a good simple experiment would be like a two-group comparison. So you have 5 treated samples versus 5 control. It's a very clear biological question that you're asking for. And then when you get the statistics, you'll be able to clearly interpret what those results mean. That you have a protein that's increased in the treated group compared to the control, something like that. Oftentimes we also have paired samples. So say they were not treated, and then the same patient was treated later, so pre and post treatment. Well, on the analysis side, I need to know that. I need to know which samples are pairs of one another. So then we're going to add that into this model matrix Lima design to account for the pairing of the repeated measures for the statistics. So there's a lot of times you'll get from me is like, what are all these different things? Tell me everything about your samples. Are they gender? Do we have male, female? What all do we have in this experiment? Because I'm trying to figure out what's going to be the best model that we need to set up to do the statistics and include. So another one that gets more complicated is factorial design. So say you have multiple drugs, you've got to knock out some sort of mutation versus control. And now you also have this interaction effect. So if it's mutated, then the drug's going to respond differently. What's that interaction? How do they depend on one another? That's another biological question. So all of these different designs are really related to what's your biological question in your experiment. This is also why we like to do pre-consults before I ever get the data. So we have clearly defined goals for the analysis. Another thing, we talked a lot about technical effects. So batching, like if you grew samples on one day and another group on a different day, whatever those technical factors are that are going to relate to a batch, we can correct for that in the model as well. You want to subtract those out of the model. So now we're looking at the biological differences and not, well, I grew them at different times, something like that. So these are all things that I'm thinking through when I look at your sample list that you send us. And that's why we kind of start with this sample list. We'll have the full list of all the samples. I'll see how many replicates are included, what factors are there. We have gender, we have drug, we have mutation. I'm going to start asking more questions about a project like that because you're starting to break the model. We start including too many things and the stats is just like, there's too many things to compute, can't do it, fail, and it won't work. So this is this discussion kind of at the very beginning. So some of the problems that we can happen, and I've had it happen before where they only had one sample in one group. You have to have at least 2. Technically, I want 5, but can't do it with 1. That's just, it won't model. We've got to be able to calculate the variance. Unbalanced designs can also be an issue. So if you've got 20 samples in one group and only 5 in the other, that's going to be an unbalanced design. It's going to be biased towards the group with the most samples. So see some of those. And some of these things are going to be hard to control for when you're doing clinical samples. Our a rare or something. I mean, not everything can be controlled for, but we do want to think about it. So controlling for batch effects, kind of talked about a little bit. Incomplete factorial designs. This is where I want to know, like, what are the sources of variability that are going to account for this experimental design? So if I leave out the batching effect, well, now that batch is going to get built into the statistical model. So you start to get false positives, false proteins that come out as significant if we're not controlling for some of the extra things in this design. Kind of one of the points I want to make about this is a lot of times if people don't like the answers of the results, they'll want to subset their data set and remove things that we know about instead of doing it a correct model that's going to account for everything. So it's almost becomes a cherry picking exercise to just, well, I want to see something different. Not the best approach to take. So just we want to try to avoid having to subset and cherry pick different samples and factors to put into the model. With that being said, we do QC to make sure what factors are going to be necessary for the model, and I'll talk about that too. Luckily, a lot of this will get recorded, so I'm posting and trying to make different YouTube videos as well to kind of try to break through. So we're going from mass spec language, topologist language to statistics now. So sorry. So overfitting, this is where you have all of these different factors that we talked about. You've got the gender, you've got age, you've got your actual group information, and you try to put all of the factors into the design, whether it's necessary or not. And when you do that, now you're starting to make it very, very specific to that set of samples, and you're going to overfit your model. So there's kind of this fine line between, do we include it? Do we not include it? What's necessary? What's not? So one of the big key things is confounding. In this case, so the image on the left is looking at, say, you have all of your females in the control group and all your males in the treatment group. Well, now we don't know if it's differences between male and female or if it's differences between the actual treatment. So what you want to do is not just randomize, but you want to do a block randomization. So have enough replicates of both of the groups in the different batches. And so this is how it's going to kind of break your statistics and have representation. And now we know the differences between the actual treatment group that we're trying to answer our question about. And this is another example like batching effect. And this was really big for TMT designs, when you could have 10 samples in one TMT batch and another 10 samples in a separate batch. When you do that, you're introducing a major batch effect between those two experiments. And so if you have 3 sample groups, we don't want to put group 1 in batch 1, group 2 in batch 2, like that. You want to randomize it, and again, with replicates within those batches. So representation of all 3 groups in all 3 batches. So I highly, highly recommend this paper if you're interested in how any of these models work. Charity Law has made a lot of helpful HTML files and just guides about experimental designs, and especially related to Lima. Matthew Ritchie, they're one of the first authors of this R package that I'm going to use. So really, really lots of information in here, and I'm going to share from this slide deck. If my mouse will come back. I'm like frozen.
I just interrupt. So it's just dealing with scientists like the ones in the room today. I mean, they've done their experiment and they are looking at male mice and female mice, young mice and old mice, plus or minus a treatment. How do you talk them out of their experiment into the right experiment, I guess is.
Yeah, sometimes it's not talking, it's well, you do want to take into these factors, but sometimes you can't overcome them, and it's just going to be in your data, and it becomes a matter of the interpretability of it. So you go into it with this knowing this is going to be discovery. You're going to find some false positives in here. We do the best job we can with the statistical model, but ultimately, hopefully that leads to a particular function, a particular biological outcome that we can follow up with better stats later or better mass spec later. So it kind of depends on what other information you have. If you have RNA-seq data, you got this other piece of data, and you're telling the story and putting all the pieces of information together instead of just everything is all in this one proteomic outcome. But yeah, my laptop is like frozen. So I may have to force close it. You have my slides over there, don't you? But I can't stop sharing. No, I just didn't touch it. So weird. There, I found a button. Maybe that'll get me there. Yeah, sweet. Okay, I'm gonna come steal your computer. 'Cause- Yeah, I'm frozen. I don't want to share that. I may have to share those later. Nothing like bread for lunch. All right, let me see here. I just may not be able to do the. I'm gonna try this again and not go into full mode. I think it's not liking my thing. So let's see if I can.
I've got it if you want.
I've got some other other things that I really would like to get to. If I can get the right resolution here. Okay, so one of the important things, and this is in that guide paper, is about regression models. And so there's two different ones that I kind of want to highlight, a means model versus mean reference model. This is going to be important if you utilize my ProteoDA package that I'm going to talk about tomorrow, how to do your own analysis. And so when I'm running most of our analyses, I'm using a means model, which means it's going to look at the means of the two groups and whatever the difference is between those groups. That's how we're calculating our p-values and our fold change information. The key difference here is in the means model, I can define which one is the wild type, which one is the mutant, and I can define the order of that fold change calculation, how we're doing our fold change. Means model reference, I can't. It's going to, whatever's listed first is going to be the reference. And so everything else is going to be in relation to whatever it's defining as the reference group. And in R, if you're not careful, it will change your groups into an alphanumeric order. And so it may not be control. It will be whatever treatment A versus control. So you don't know really the order there if you're not careful and you don't set that correctly. So I prefer to do the means model. I can clearly define what my groups are. I can clearly tell it, I want these different comparisons and run all the statistical information. So one of the things that's really important is this design matrix and contrast matrix. When I'm talking about factors that we're going to add into our experiment, this is where it comes into play in the statistical side. Each column is going to be a group. So control treated. You have two groups in this one. If I had a gender effect, so male, female, that would be an additional column or an additional factor. And so these are going to be all the things that we're trying to explain by this linear regression model. The rows are going to be the individual samples, and that's how we're flagging it as belonging to the control or the treated group and what the different characteristics are for the samples. So if I have more factors, more columns here than I have rows, I don't have enough replicates to actually do that model. So if you keep this to a 5x5 experiment and we've got 3 replicates, it can handle that. If you do controlled, mutated, 3 drugs, 2 time points, I now don't have enough rows to handle all of that variability and all those different factors. So that's kind of where the model just starts to break down. There's just too many different definitions, too many factors, and not enough samples to handle it all. So when we're doing clinical samples, a lot of times you want, 10, 15 per group, or, even more if you're looking for biomarkers. So everything is going to depend on your biological question, your ultimate goal of your experiment. And so that's why pre-consults are helpful. So the contrast is going to be, that's just the definition of telling it what your groups are. You're defining what we're going to compare, control versus treated or whatever that is. But it's the language of the statistical model. We have the design and the matrix. So we're using this no intercept model. The no intercept model means have this tilde0 in front of this model matrix. This is the actual code. And this is what's really important if you use my R package tomorrow. If you don't have the 0, you just do tilde group. Now it's going to be that mean reference, and it's going to put the order however it alphabetically defines the groups. So that's how we're going back into linear algebra, pretty much. So this one is just an example of having more treatments, more factors into the model, but you also want to have more replicates that are included in that data. So if we had like a technical factor, a batch factor effect in here, it would actually add that into this design as a random error term that will help adjust all the stats and remove batching or whatever it is we're trying to remove from the model. So this is one thing that I see a lot, and I wanted to bring it up because you want to be careful of sample terms and identifications. So what has happened here is, say we have a TMT experiment and we have 2 sample groups, A and B are in the first TMT experiment and C and D are in the second experiment. Now these are confounded and we don't have full rank. And so what it's going to do is it's going to try to estimate the mean of batch 1, the mean of batch 2, and I can no longer subset out A and B, C and D. The model just doesn't know what to do with it because it's a redundant label of that sample. And so these are the things that when I see a sample list that I'm trying to kind of watch out for and make sure I don't break the model at the end. And it can get really crazy when you start going in interactions and stuff, and I'm not going to go there. I just put a lot of text here from that outcome, which you can have. So how do we know what should be included as a factor in our model or not? This is where our QC plots come into play. After we've normalized the data, we've got the data ready for analysis. We want to look at dendrograms, PCA, MDS, any other type of plots that tell us if samples are outliers, if there's a major separation between males and females, and what all do we need to actually include in our model. So I did want to bring up an example data set. And so here is an example of, these were primary cell culture samples. And what we're looking at for each of the samples is just the total intensity. What's the total intensity of all the proteins identified for each of those samples? And you can clearly see some samples did not have much protein detected in them at all. And so if you just kind of look by batching across different things, different sets of samples had less protein than other sets. And so those become problematic samples in the analysis. So we looked at 15 samples out of, I forgot how many were in here, a lot. 80% missing proteins for those 15 samples. Those are going to be outliers that we then end up just having to remove from the analysis entirely. And you can see it in a lot of the PCA plots. So here we're highlighting those 15 samples in red, and you can see that they are sort of separated out at PC1 and PC2 in our PCA plot. So one thing about PCA, a lot of people, I think, this is just my opinion here, kind of overutilize PCA, because especially our assumption is the majority of the proteins don't change. And so if the majority of proteins are not changing, that's what PCA is looking for, is separation variability. Your sample groups may not separate on PCA, and that's okay. I'm looking for things that I know would be a factor, like we dropped this sample on the floor, or there's some biological reason that has happened before too. That I'll have some sort of information that says, we really do need to throw this out. This is an actual outlier. This will help kind of identify those samples, but I usually don't just throw them out, because if you include it into that model, that helps with the variability estimation, and it actually gives you more power in your model. So I'm not a proponent of just throwing everything out because it's over here in this corner. So, and we're only looking at, usually I get maybe 20% variability across PC1. This one's actually a lot more, but this one's going to be a lot more because I just showed you on the intensity level, we're missing like 80% of the proteins. So that's a justified reason those should be removed. So yeah, we kind of colored by, these were also, I think, they were done in different blocks, which is kind of like a batch. They just called it a block. And so if you start coloring by block, you can start to see some of the blocks are, yeah, they're clustering together. So in this particular model, we'd probably want to include that blocking factor into our model, because we do see separation by each of those batches of the samples. So if you look at just the overall intensities, we've got these samples down here, they're just causing problems. Those are gonna just be separated out and taken out of the overall analysis. And you can see it again too with the dendrogram plot. Get rid of my Zoom thing over here, see if I can, oh, I may have closed it. I think I did. I closed that one. This is the outlier samples. So if you're clustering by similarity, those outlier samples tend to be over here on the side of the plot, kind of also giving you information that they're separated out. Okay. Does anyone have questions? Yeah.
Sure.
Yes. Yes.
So, you know, I like to think about, well, you know a lot more about statistics than I do, but I like to think about it. I mean, in the end, you can have a lot of variables and you can obviously process a lot of samples.
Yep.
And then it seems like, okay, I mean, you want to go in with some ideas, but you can kind of see how the data falls out and decide to make easier comparisons than everything at once and sort of complex models with multiple variables. I mean, would you say that's fair?
Yeah, so we've done this before too. So if you see like there's a major difference between males and females, and you're certain, you just do the pairwise comparisons where we're compare everything in the males and the females separately. And that's really important for certain studies because all of their stuff has to be done that way. They're looking at hormones or something, and that's very gender dependent. So yeah, it always kind of comes back down to what's our ultimate goal and what type of samples are we dealing with. But yeah, I've done that before, and usually on something like that, you'll get multiple analysis reports where I'll do it with it included in the model, then I'll do them separately by both. And then you kind of looking for what's unique and because if I include sex into the model, then what that's going to do is tell you these are the major differences overall, regardless of the gender, between your treatment effect. If you don't, and you do it separately, and now you've got males, females, separate analysis, control treatment, you may have some unique proteins that show up from that analysis in males that don't show up in the female. And that's going to be a biological insight that is going to help you with your research, say, hey, these are very unique to this subset of population or something. So yeah, it's all coming back to, so what's your experiment?
So when you show the mouse head categorization, you had 4 mice, and then you had 2 males and 2 females in each group.
Yeah.
So again, I wanted to know, like, you set a standard of 5 samples. And in this case, you're using 4. And then if you were to look at the variables, they are even less, like 2 per treatment set.
Yeah, so the
how do
4 is not, this is just somebody's example to understand what confounding variables is. But here you could do like 3 of males, 3 of females in control, and then 3 in the other group. So that way you've got at least a representation of all. I'm always a proponent for more samples. So that's also why we try to make our proteomics cost not reflect the fact that I want more samples. So, yeah.
So let's say you have an experiment. It's a transgenic model versus a wild type. You get
Yeah.
10 animals in each group, 5 males, 5 females. It's great, but it's a longevity study. So let's say only 7 rats survive in your transgenic group. Would it be best then to randomly remove 3 of the wild types that you have 7 and 7? Or should you still look at all 10 of the wild type and the 7 from the transgenic?
I would run all of the samples on the mass spec because I can always take one out in the analysis later, but I can't get more data. So if there's like a bad sample in one of those 10, that's going to become an outlier. But 10 by 7, well, you can usually overcome the unbalanced. Yeah.
Is there a model other than Lima that could be applied, or is it not? It's not worth?
No, not for that. Lima is really the best one. Yeah. So, yeah, it's like
Is the batching effect something to consider with DIA proteomics, since all samples are being run one at a time?
So that's the good thing with DIA. When we do just regular label-free analysis and you're injecting a sample every single time, well, now you're going to introduce a bunch of zeros in each of those samples. With the DIA, I tend to have less missing data because it's going to try to sequence everything in that same window. And so I typically don't have as much issue with the DIA for that. Yeah. Which is why I like it. So I'll probably steer you towards DIA almost every time. Yeah.
It's also cheaper than TMT, so that's good.
Right. And I can get more samples.
Another question actually about batching effects for cell culture. I know we want similar confluences in order to minimize any differences. What are some of the other things that we should consider? Should I be getting all of my N's from the same flask?
Yeah, so that's a great point. And so this is the other thing that I try not to overthink is trying to make your experiment too sterile. Because if you make everything very, very sterile, you get a certain result, certain answer, that experiment. You do the same experiment a few months later, you may get different answers. And so for cell culture, one thing that you can think about is, say, you're going to grow up cells that are controlled treatment, all of your different sample groups within that time point, that batch, same media, everything for that. Then you could do it again with a different batch of media, different flasks, different everything, but same groups. And that introduces a little bit of variability into your cell culture that you normally won't have. So if you prepare everything the same media, same flask and split, that's more of a technical replicate than kind of a biological. So yeah, great question.
So we do want, at what point would that introduce the batch effect so we don't really want, yeah.
Yes, so that's something we're going to look at in the QC. You'd make a note, these are all done together. These are all done together. And hopefully when we see the PCA, there's really not much of it shouldn't be the strongest factor. Your strongest factor should be your treatment or your main effect. Yep. But good points.
Can you use DXSeq instead of Lima here?
Not here because those are based on count data and we're working on intensity data. So Lima has ways to handle intensity type input. Yeah.
So DXSeq 2 is only for the count data.
Yeah, it's only for count. So it's RNA-seq type data. Yeah.
Thank you.
EdgeR's EdgeR's got some functionality that we actually pulled into here too for some of the outcomes, but the model itself is Lima. Yeah. And that's something I do a lot with RNA protein integration, because you are going from counts to intensities. But if you can use Lima Boom on your RNA-seq data, and that will turn your counts into an intensity-like matrix. And now you've got a similar data type to work with. So. Yeah, I'm interested in multi-omics too. So if anybody wants to talk offline, we can do that. Okay.
So what I hear you say is we want to measure the variability
Yeah.
and we don't want to artificially limit the variability.
We want biological variability, not technical. So what I'm trying to remove with normalization is going to be, say, loading one sample on the instrument and then another one after that. The instrument's going to introduce some bias and some variability itself. So the normalization is trying to remove anything that's artificial, technical. Yeah.
Can you use some kind of spike in to normalize?
We don't for this. I have in the past for like CELD data. That's like way ancient. Yeah, there's a bunch of, so your bioinformatic methods tend to work off of yeast, human cell lysate with spike in so that we have a known control to figure out what the algorithms are. But we don't currently do that on a regular basis. And tomorrow, I'm going to bore you to tears and actually show you our code of how to run your own analysis. So, and again, I did record a video on this, so it's already on the Arkansas Embry YouTube channel. The point of that is to be able to put this power into your hands. So do you want to change a group comparison or something? You have the opportunity to do that without waiting on me. And so I am going to kind of walk you through that demonstration. And I think as I go through that in each of the steps, probably more of this type of conversation should happen because I'll remember something else. But so you can be thinking of that too.
And I'll make a plug, even a chemist can do this.
Yeah, I taught Dennis.
Yeah, she taught me and I was showing Joe my stuff yesterday, and I said, now realize that this is like, I know how to get in the car and turn the key and drive someplace, but if you open up the hood and say, what's that? I go, I have no idea. So she's going to explain all of the what's underneath the hood sort of thing, but to actually do the script is actually not that bad. So,
well, thank you. So. But yeah, and we're willing also, if you get a data set, we always do a post consult usually with people and kind of walk you through specific questions. This all comes back together when you have your own experiment and you know what it is you're looking for, then it makes a lot more sense. So.